And hello everyone. It's great to be here. Let me see. Let me just share my screen, and then I will start just a second, like that. Okay. So the title of my talk is "The Physics of Stinging," which is, as Andreas pointed to, a paper that came out recently. It's actually a project that started, I think, 3 or 4 years ago. And so I will start with a little bit of background into why we started studying the physics of stinging and provide a little bit of context to some of the background in the field before we state what is the main research question. So I'm a physicist working with plants. And I mean, it's not necessary to justify having an interest in plants, but let me do so anyway. So, of course, in terms of biomass, it is the largest group of living organisms on the planet. It's important for food, for you and I, and also in terms of energy, it is a not only current source of energy, but also historically a very important source of energy. However, if I'm to characterize plant life in just 2 words, it is really that they are under attack. So if you think of a plant, a very fundamental condition is that it cannot move on timescales comparable to what animals are able to do. So if you are a plant, your main experience will be this. So someone is constantly trying to eat you, essentially. And plants use a wide range of strategies to defend themselves against herbivory. The main strategy is chemical defenses. So plants have extremely refined organs and synthesis pathways for producing and releasing phytochemicals that will deter certain herbivores or attract insects, for instance, to defend themselves against other living organisms. So chemistry is the main weapon in the arsenal, but a few plants use some interesting physical mechanisms to defend themselves. And so I would like to highlight one, which is, at least it's found in my backyard, which is a plant in sort of the nettle family, stinging nettle. Which has these sort of characteristic jagged leaf shapes, and the leaf surface and the petiole and the stem of the plant is covered in these hair-like structures that are little stingers filled with poison. And so this strategy, poison-filled stingers, is used by approximately 1% of plants. And so this particular species were very widespread in Europe. You also have sort of relatives in Australia, South America from the same family. So it's a widely used strategy. So let me highlight some of the features of this particular envenomation strategy. So the stinger is an approximately circular cylindrical tapering cell. So this 1 millimeter long stinger is a single cell. It has a cell wall which is reinforced by minerals. So you can simply think of this as being made out of glass. It is filled with poison. So this entire cell is filled with poison, and then there's also a poison reservoir down at the base here. So important features are that the stinger up here itself is rigid. It's relatively stiff, at least, made from glass, essentially. And then there is a soft base, which is flexible. And the whole thing is, the poison is kept in the stinger by a little spherical cap. Okay, so we are not the first to ask the question of how this stinging or poison injection strategy works. So the first that I know of, at least, is Robert Hooke, known, of course, from Hooke's law in elasticity, but also for inventing the compound microscope. So this is Robert Hooke's drawing of the leaf surface of the stinging nettle. And again, here you see the highlighted features are the basal flexible structure and then the glass-like needle here. So Hooke's stinger model is quite simple. So if you touch the stinger, it first comes into contact with the with the skin. Then the cap breaks off, and as you then move relative to the leaf surface, where the base here is stuck, the poison is released. So I went into my backyard to try to see if I could replicate this experiment. And so these are some experiments that I will show now on the stinging mechanism. And this over on the right, this is my finger. The length scale is approximately 1 millimeter. And then we mounted this sort of stinger on a micromanipulator. So let's see what happens. So before I describe what is happening here, I need to make sure that you can see the video clearly. Is it good? Okay, let's play it again. So you see the stinger when it comes into contact with the skin. The stinger, the glass reinforced cell, remains straight, while the base structure allows the needle to pivot, and it pivots almost to 90 degrees. One can be stung by multiple needles at once. This is quite painful. I should highlight that this is not an experiment that I recommend you try to replicate on yourself. And we have also done experiments on, let's say, artificial skin. So this like a gelatin product with a thin coat of molten paraffin. So it's a paraffin layer. And this over here is the stinger. And what you can clearly see is that as it penetrates, it injects quite a substantial volume of fluid below the surface here. And I don't know if you can see, but one observes the receding meniscus, which moves almost to the base of the stinger. So it's almost completely empty from fluid. Anyway, we have now, this is our first observation. It's a slightly refined model of Hooke's poison injection mechanism. And we have identified this, let's say, the rotation as a key element of the poison release. And so if you work in a chemistry lab, you're familiar with these, let's say, pipettes. So this is really how the stinger actually operates. And the volume which is expelled from such a pipette scales approximately linearly with the angle. So it is quite a, let's say, it's a very simple strategy for injection of poison. So now I want to step back a bit and think about other stinging processes in nature, because what we observed in the stinging nettle is that it's important, or at least for this poison injection mechanism, or for this stinging process, that the poison-filled reservoir here remains straight. Otherwise, this pivoting motion cannot inject the fluid. So for this reason, we started to take a broader look at stingers across both nature and technology, just to understand how the interaction between the mechanical function and the shape and morphology of these stingers are connected. So if we start on the left over here, I'm showing one of the smallest stingers that we identified, which is a lambda-phage. So it's a virus that infects bacteria. And it has stingers that are approximately 100 nanometers in length. And they are used for adhering to the bacterial, let's say, bacterial surface before the virus penetrates the cell membrane. Now this axis down here, I should point out, this is the characteristic length of the stinger, which is in units of meter. So we go from 100 nanometer up to around 1 meter, a little bit less than 1 meter. So it's a soldier wielding a spear here, so it's a man-made structure. But in this range, we find both a wide variety of living organisms. So everything from algae to plants, insects, land animals, marine animals, and also man-made structures such as needles, nails, and weapons are all found in this range. And so let me just give a few details about some of the organisms here. So here you see, so this is an SEM picture. and again of the lambda phage, and it has successfully made the touchdown. It looks like a little lunar lander. You can see it made the touchdown on the bacterial surface, and now it's adhering using these spikes here. Another example is the wasp here, where the stinger is about to penetrate the surface of the skin. And more exotic example is the narwhal that has a tooth which grows to this extreme length. There are many, many interesting things about the narwhal. It is the longest stinger in nature, perhaps up to 3 meters, the longest extant species that I know of, at least. It's not clear that it's used as a weapon, actually, but that's a different discussion. Maybe we can elaborate on that a little bit later. I want to point out that many mammals and also land animals in general use the stinger-like structures, let's say weapons, in a social context. So for in competition for mating, essentially, they grow extreme stingers. So this is the Iberian oryx, or the Arabian oryx, sorry. And this over here, I believe, is a moose. And you see they invest substantial resources into the formation of these outgrowths, right? So it's not only for use as a weapon, but also for, let's say, social interactions. They are, however, deadly. So this, I reported in the Washington Post from 2019. At least a hunter was apparently gored by the deer. Just to point out that it has a potential to function as a weapon. There are also, I mean, the marine animals, so these like swordfish, they actually don't use it to pierce, so it doesn't move typically perpendicular to the prey. They use it for a sort of a slashing motion, so from side to side, in order to stun the prey. They're not always able to capture the prey, as this video illustrates, of course, but that is what it is. Okay, so now just to, let's say, confine the discussion, because this can become incredibly broad, I will only talk about, let's say, straight, rigid stingers that are slender and taper approximately linearly. And we look mainly at stingers that have circular cross sections. There are some examples of different shapes in nature, but this is really the dominant shape. And the process we study is this stinging, a tapering rod of base diameter d0 and length L, trying to penetrate some target tissue. So this could be the human skin or the surface of a bacteria. It's a target characterized by some parameters that I will cover in a minute. And so now to our question. So if we look broadly at the landscape of stingers, we note first that they vary in length from 100 nanometers up to about 1 to 10 meters. And they also vary quite substantially in, let's say, thickness. So both length and thickness vary. And of course, the question of how these two are correlated. So for the stinger to function, how wide must it be if you require a certain length? And if we look at the data, we clearly see a linear relationship, so a one-to-one relationship on this logarithmic plot here between length and diameter. And I note here that, so there is an analysis of nails. So this particular example up here with a hammer driving the nail some time ago by McMahon and Bonner, which predicts a 2/3 power law for driving of the nail. And as you can see, the data doesn't actually follow the 2/3 law. So the central question here is, what is the physical design principle behind this one-to-one relationship between length and diameter? So that's what we'll try to address today. Our fundamental hypothesis is that the mechanics of stinging is limited by two physical phenomena. So for a stinger to function properly, it must be able to overcome the friction in the tissue. So as the stinger penetrates the target tissue, it experiences a friction force, So the force you apply in order to penetrate must exceed the friction force, otherwise the stinger cannot penetrate any deeper. While the force must be sufficiently large to overcome friction, it is generally favorable if the stinger does not buckle. So if we can avoid mechanical failure, and here I characterize the mechanical failure simply in terms of Euler buckling. So if the force exceeds a certain threshold, we have this characteristic very rapid transition between the straight geometry and to the bent. So we'll cover this more in detail later, just to give you a qualitative picture. So again, we must overcome friction and avoid buckling. So we can formulate the design principle in a relatively simple way as an inequality. So the applied force must exceed friction and avoid buckling. So let's look at this buckling process first. So what our approach was to try to build artificial stingers. And in order to study the effects of elasticity, we made rather soft stingers. So these stingers are made from a polymer material, PDMS, frequently used in, let's say, microfluidic device fabrication and so on. And here I show one of our experiments. So the stinger is mounted on a load cell. And this up here is a linear translation state that can move up and down. And as you can see, the stinger here is relatively large. It has a base diameter of approximately 1 centimeter. I should point out that this movie is, I mean, the experiment is done, let's say, quasi-statically. So this is sped up around 100 times or so. Okay, so it's in reality, it's a very slow experiment. But you see this characteristic transition between the straight morphology and the buckled geometry. So now the question is, what happens when you try to make the stinger more of a stinger? So it has to have a slightly pointed geometry. So what we did was to take the same mass or the same volume of material and make it more pointy. So here you see a taper, which is, let's say, approximately 2 to 1 in terms of diameter ratio. And the basic observation is that it fails at a smaller load. Okay, so the stinger is able to withstand a smaller force than the straight column. And again, if I do the extreme, I think this is like a 20 to 1 in terms of this base diameter to the tip diameter. You see that it actually fails almost immediately. And also that while this buckling gives you, very simple, let's say, symmetric deformation, the buckling process in this case is confined almost exclusively to the tip. So while the base here pivots, it stays relatively straight. So we did this for a bunch of different geometries, and let's say if I plot here, so this is the ratio of the tip to base diameter. So on the top right-hand corner up here, we have the straight cylinder, and all the way down here, I have the most sharp stinger. That's on the horizontal axis here, and on the vertical axis, I plot the force required for buckling, normalized by the force required for buckling of the straight cylinder here. So you see this sort of plateau-like behavior on moderate tapers, and then a very strong decrease in the force required for buckling as the stinger becomes more and more pointed. So of course, the question is why this particular behavior? And so we can do a very simple modification of standard buckling theory to include the effect of tapering. So for a cylinder of constant radius, we have sort of a, let's say, a balance between the applied load and then the bending, which we characterize here simply by the, let's say, the curvature of the rod. And for a straight beam, we find this particular beam equation, right? So the parameters in here, so this is the applied force, it's Young's modulus, and we have the moment of inertia. And the moment of inertia, of course, depends on the beam geometry. So it is essentially the area squared. And so the critical load for this beam geometry depends on Young's modulus, moment of inertia, and then the length squared is in the denominator here. If we look at a linearly tapering cylinder, the beam equation is essentially the same, except for the fact that we need to replace now the moment of inertia is no longer constant along the beam. So it is, let's say it's larger down here where it's wide compared to the tip where it is smaller. So it is locally, it's much more unstable up here close to the tip. So the moment of inertia is now position dependent. And as long as it varies relatively slowly, we can simply put that into the beam equation here in the denominator. So you see the differential equation is now slightly different. And we find a different critical load, which now depends on these geometric parameters. So essentially how tapered it is needs to be factored into this equation. So if we take this solution to the beam equation and we plot it on top of the data, we find a nice agreement between this simple model of buckling and our observations. Okay, so back to our design criteria. So the design principle for the best stinger is again that the applied force should be smaller than the buckling force. We just saw that this depends on the stinger geometry, how it penetrates, and the material parameters. And the second component of this design principle inequality is then the friction force. And as a stinger penetrates a tissue, during that process is of course, that process is of course a very complicated mechanical and biological problem. So there many parameters enter into this. We model it in a sort of, let's say, using perhaps the simplest possible model. So we simply say that the friction force experienced by the stinger is the integral over the embedded surface. So the embedded surface would be this surface area down here. And then it is a friction coefficient. So the friction coefficient between the, let's say, wet porous tissue and the stinger surface multiplied by the characteristic tissue pressure. And then we integrate over the full area. Now the tissue pressure we can estimate based on physiological measurements. The friction coefficient we can estimate based on experiments on, let's say, intrusion experiments or penetration experiments on wet porous materials. And so if we carry out this integral for a simple linearly tapering stinger, we get this dependent. So we note that it scales linearly with the tissue pressure and the friction coefficient, and it scales linearly with the needle aspect ratio. And then it's quadratic in the penetration distance. So the deeper we get, the higher the force proportionally becomes, which also is in accord with experiments on actual, let's say, hypodermic needle experiments. Okay, so if we go back now to our design principle again, we have these two characteristic forces. So we must never exceed the buckling threshold, which is characterized by this expression. So the buckling force depends on the elastic modulus of the stinger, the penetration depth, the diameter of the needle to the power 4, and then the length to the 4th power. We must always overcome the friction force, which I just discussed. So what I plot over here on the right is the diameter to length ratio, normalized, probably normalized, and then I plot the force here on the vertical axis. So the dashed line, here's the buckling threshold. So essentially, we can never be up here. We can never be above the line, then our stinger will fail mechanically. We can never be above the dashed line. Similarly, we always need to be above this straight line here, which is our friction law. So it scales linearly with d naught over L. So we always have to be up here. So you see the only permitted region for our stinger to operate is this blue area up here. And if we look at a stinger of a constant length, say your organism needs to penetrate the tissue of a characteristic depth, or it is attacked by a prey of a characteristic size, then the cheapest possible solution will lie right here. So the thinnest possible stinger you can make will be characterized by this intersection point between these two force curves. So in nature, you want to live here. Evolution will favor that. If you want to apply this to technology, it might be a good idea to move, let's say, slightly upwards here. So you also have some safety and resilience. Anyway, the optimal configuration, this dot, is characterized by a linear relationship between diameter, stinger diameter, and stinger length. Then it depends on the material parameters, of course, so the friction coefficient, the tissue pressure, and the elastic modulus. But these enter into the expression only in the 1/3 power, meaning that the dependence on the particular tissue characteristics or the particular material from which the stinger is made plays relatively little role. So now if we go back and look at all the geometric and material data from the stingers we discussed earlier, we can try to ask the question if they actually adhere to this design principle of the optimal configuration in terms of function and cost. And that is what I show in this plot. So if we look across all the organisms that I discussed earlier, we see that they scatter nicely around this solid black line, which corresponds to the expression over here on the left for the optimal design. So I would say there's broad agreement over these, let's say, 8 orders of magnitude in normalized length and diameter. Now, it is worth highlighting a few outliers here. So algae, so these are silicoflagellates, they use the stingers for 2 distinct functions. So one function is to prevent grazing. So in order not to get eaten, they will grow very long stingers. Simultaneously, they also use them for, let's say, staying afloat in the water column. So to increase the sedimentation time, that is also a reason why they produce the stingers. So successful tissue penetration is not the only design criterion. I should also point out that they are hollow. So for a hollow stinger, all things being equal, it needs to have a larger diameter in order to have the same mechanical characteristics. Let's say insects are interesting in this context. Also, you see that there are quite a number of insects that fall below the line here, so that they are actually softer or will buckle more readily than one might expect. And one interesting example is in the wasp family, where many wasps use the stingers not to pierce the skin of animals, but to lay their eggs deep inside wood. And during this process, it's very important that the stinger is flexible and able to bend inside the wood. Finally, let's say nails and needles here. When you drive a nail, inertia is most likely a very important effect, which is not included in this analysis. So it is entirely justifiable that they appear to be slightly over-designed. It's not unexpected. Nevertheless, if we look at the broad picture here, I would say there's rather good agreement. We are able to at least provide some insights into the reason for this one-to-one relationship between diameter and length. I think that is what I have to say. So I thank you for your attention. I hope that we can, I will be happy to take a few questions and we can have a nice discussion. Thank you.
Okay, thank you very much, Kaare. Do you have any questions?
I have a question, Kaare. Thanks
Yeah.
for the talk. Very interesting. In your relation, it seemed to my eye that the cloud of points was systematically up. So D0 tended to be larger than what you would predict by the one-to-one relation. Is that right? And if so, what are the parameters that would be responsible for that?
Yes. Okay. So this is a very good question, whether or not it is systematically up. I mean, I should point out that what I plot here is actually, I mean, there are no free parameters. So even this 32 over pi squared to the power 1/3 is in here, okay? So it is perhaps not unexpected that there will be some systematic shift. So I think that some of the effects that are important is that we assume that the stingers are characterized by a uniform medium of a single Young's modulus, whereas in reality, many of these stingers are composite materials. So they will have a, let's say, a stiff outer shell and then a softer inner material or foam-like structure inside. So I think the composite nature of the materials might be important in understanding. It might also be a question of resilience. So if you systematically make your stingers too weak, like a too high proportion will not function properly. Okay, so maybe they're a little bit over-designed. Maybe nature will favor that. Does that answer your question, or do you have other ideas?
No. I think the second explanation makes sense because you assume you are at that critical point, right?
Yeah.
So that's not necessarily the best solution in terms of resilience, as you point out.
No.
But that would actually move it up.
Well, I mean, like if you assume that the function is dictated by the length, then you can move vertically in this diagram, and the higher you go, the more stable you are, but the more expensive it also becomes because the volume you need to invest, or the mass you need to invest, increases.
Yeah. Thanks.
Jorge Cuadra has a question.
Yeah.
Go ahead, Jorge.
Hi. It was a very interesting talk. Thanks. I was wondering, you mentioned that for nails, it has been proposed that they should have a 2/3 slope in this plot.
Yeah.
So could you explain how one would get that slope and whether you find any support for it? Because looking, as Andres just said, here I would say all nails and needles are a bit shifted above the curve, but I don't see any other slope.
Yes. So the reason for I think it is a little bit more because everything here is also scaled by Young's modulus and so on. So one needs to be a little bit careful about. If I can just go back to the original plot where maybe we can see the 2/3. So that the 2/3 line is here. I mean, the data are statistically closer to 1-to-1 than they are to the 2/3. Okay, so we tested this. But if you just pick out the nails up here, 2/3 is not a bad approximation. And the original analysis is based on the hypothesis that all nails will buckle at the same characteristic penetration length. So no matter if it's a 9-inch nail or a tiny nail, it will always buckle at the same penetration length. So in that analysis, this whole idea that friction increases as you penetrate deeper into the tissue is not included. That's how you get the 2/3. But it's a beautiful, I mean, I would highly recommend to you to look in this book. I think it is online. If you just Google this on size and life, I think it is available online.
Thank you.
More questions?
I have a question, Kaare.
Yeah.
Here, essentially everything more or less in nature is kind of a adiabatic, so all motion is slow. But if you have for example, an arrow, or you have something that you have something that is very fast, so you may activate this high order modes of bending. So have you looked into that?
So on this plot, or in our data set, we actually have a few arrows, and they are typically they fall below the line, and I believe there is some evidence that this oscillation, as you say, this higher order, let's say, mode, is important for flight stability. Or at least it doesn't prohibit the function of the arrow, right? I think the crossbow bolts are typically have a larger diameter, and they are closer. They are closer to the line.
Perfect. Thanks.
Yeah. But excellent question. Thank you.
Any more questions? No? Okay. So, well, thank you very much, Kaare, for this very
you're
nice
welcome.
talk, very interesting. So, thank you again, and also thank you for being from far away and giving this talk to us. So, if you guys want to, keep hanging out, we can stay. But if let everybody go free if they want to. But let's just finish here. So, thank you.
Okay.
Again.
Thank you so much for inviting me again.
Thanks
Pleasure.
for the talk, Kaare. Very
Thank
nice.
you.
Thank you,
Thank you.
Kaare. Thanks a lot.
All right. Okay, I'm signing off here. Nice to meet you, everyone. Bye-bye.
Okay,
Bye.
thank you again, Kaare. Take care.
Bye.
Take care over there.
You too.